I'm a professor in neuroscience at the University of Oslo. I've been, I did my medical degree at the University of Oslo and did my PhD again in Oslo. I did part of my postdoc at Harvard, and now I'm back in Oslo, and since 2000, I think 12, been working as a professor. So I'm going to talk about brain aquaporins. But first of all, you asked me to say a bit about my journey. So I'm born in Kerman, as you see here, and now I live in Oslo. It's actually two places with lots of contrast. I don't know if any of you have been to Kerman. It's I like it a lot. It's rather dry, many sunny days. It can get cold, but Oslo is not dry at all. It's also cold sometimes, but it's much darker than Kerman. Otherwise, I mean, I don't mind living. I like being in Oslo in Norway. I love the country. I was also exchange student in Italy for half a year during my medical studies. And this is the first day I started my medical studies. I don't think you can recognize me, but if you do, that's me. I'm sitting there. My brother and sister also started at the same time. You know, when we left Iran in the 80s, and I was the youngest, but you know, when you come to a new country, you reset everything. So from being the youngest, we started going in the same class. So I think it was a lot of fun. At the time, I was studying medicine. This is the book. It's actually my book from that time. It's from 1991, and Human Physiology. I don't know if you have read this book. At that time, this is basically all I found about how water crosses the cell membranes. Our understanding was that basically what is written here, that many substances can diffuse freely through the lipid membrane, especially water and dissolved gases. And then there was something else about the kidney, the water transport in the kidney, where it was, I mean, a hint to probably water permeability can be regulated by antidiuretic hormone, ADH. But there were no aquaporins in my physiology book. At the second year of my medical studies, I started doing research with one of my professors. And at that time, I started working on kidney. And at the same time, more or less, something was going on here in the US, or where you are right now. So this is our understanding of water. Basic diffusion through the lipid bilayer. Peter Agre, who later became a very good friend and collaborator at the Johns Hopkins University School of Medicine, he was working on red blood cells. And in it's written 1998, it's supposed to be 1988. His group, they were looking at rhesus factor proteins, and while looking at them, they noticed a consistent contaminating band on their gels. Those of you who have who are familiar with running gels, you know that very often we find bands that we don't like or we don't want them to be there. So instead of just forgetting about that band, it caught his curiosity, and they started looking for it because it was there all the time. So what they did, they actually purified it, they raised antibody against it, and they found that it actually a large portion of all red blood cell membrane proteins is actually composed of this protein, which had a molecular weight of 28 kilodalton. And they also found that it was abundant in kidney. Later, when they cloned it and they expressed it in xenopus oocytes in 1992, they actually demonstrated the proof that this protein was a water channel. So before that, at least for most people, the concept of water channel wasn't known. So what they did was, when they expressed this new protein in xenopus oocytes, they saw that in a physiological solution, nothing happens. But when they move these oocytes to hypotonic solution, within 2 or 3 minutes, these oocytes start swelling, and finally they burst. And that was the proof that this is a water channel. You know that xenopus oocytes, they are not permeable to water because these frogs, they lay their eggs in fresh water. So these they have to be resistant to water flux. And so that was the aquaporin, and this is Peter. He's with his group, and that's the discovery of aquaporins. And how can a channel be selectively permeable to water? That was also a question. Later, the atomic structure was described, and so the reason for its water selectivity is partly size restriction, that the narrowest part of this channel is large enough just to let one water molecule to pass through. In addition, there are some positive charges inside the pore, which repulse protons, H3O+. So that combination of size restriction and electrostatic repulsion makes these channel selectively permeable to water. So what we today know is that water can cross the lipid membrane both by diffusion, but also by pore-mediated flow. So these water channels, they mediate rapid water flux. There are also some other molecules like co-transporters, like sodium glutamate co-transporter or sodium glucose co-transporter, which transport significant amount of water as well. So our understanding has changed. So Peter Agray, actually 11 years after that discovery, he was awarded the Nobel Prize in chemistry in 2003, and that's he's on his way from Stockholm. Actually, we joined him in his Nobel party, and he's actually he is a role model. So besides being a brilliant scientist, he is also engaged in what is happening in the world around him. After the Nobel Prize, he was director of a malaria center, doing research on malaria, and was very often in Africa. He has been also involved in science and diplomacy. So he's absolutely a role model. So today there are 14 members of aquaporins in the mammalians, and more than 200 in plants and microbials. So, so this is a very well preserved family of proteins throughout the evolution. And aquaporins, I mean, this is an old figure, so not all members are written, are shown in this picture. But we have two main families, one aquaporins, which are mainly permeable to water, and then aquaglyceroporins, which in addition to water are also permeable to glycerol and some other solutes. I'm going to focus on only aquaporins and brain aquaporins. So in brain, we have three of them, which are expressed both as mRNA and protein. Aquaporin 1, which is in choroid plexus, where cerebrospinal fluid is produced. I'm not going to talk about it. Aquaporin 4, which is the predominant aquaporin in the central nervous system. That's the focus of talk. And aquaporin 9, which is expressed in the subpopulation of neurons and astrocytes and probably some other cells. We are still doing some work on it. It's not resolved yet. So my talk is going to be on aquaporin 4. So aquaporin 4, you know, being a neuroscientist, I know that most people are focused on neurons, but maybe not. Increasing number of scientists are interested now in other cell types in the central nervous system, such as glial cells like astrocytes. That's what I do. My focus is astrocytes. And, you know, astrocytes are one of the most abundant cell types in the human brain. They are star-formed cells which, with food processes, so-called end-fit, which surround all microvessels in the brain, and I mean in the CNS, or the brain surface and spinal cord surface is completely covered with these processes. In addition, they also have processes surrounding neurons and synapses, and they communicate, they constitute a network through gap junctions. So they fill astrocytic processes, they fill most of the space in between the neurons. And a colleague of mine, Alan Nagelhus, this is the immunogold picture from him. You probably are familiar with immunogold. It's like immunohistochemistry by electron microscopy. So the principle is the same, that you have a primary antibody and secondary antibody coupled to a gold particle, and these gold particles are shown as black dots. So here you have an endothelial cell, it's a brain capillary, and you see these black dots are on the astrocyte membrane facing the vessel. And here is the brain surface. Again, most of the highest density is on the end fit facing the vessel or the PL surface. There are some in astrocytic processes surrounding the synapses, as shown here is the glutamatergic synapse. If you look at this astrocytic end foot with another technique called freeze fracture, that's how it looks like. You see these complexes of particles here. These are all aquaporins. These were actually discovered several decades before discovery of aquaporins. People didn't know what they were, but you know, they fill a large portion of the astrocytic membrane facing blood vessels. And these are called orthogonal arrays of particles. Aquaporin 4 knockouts, they don't have these particles. So it's a very, aquaporin 4 is a very abundant membrane protein, which is localized highly in a highly polarized manner at the astrocytic food processes surrounding vessels and brain surface. So one of the first questions we asked was that since aquaporins are exactly on the interface between blood and brain, The question was whether this is the entry route for water to the brain during brain edema formation, and also exit route. As you know, brain edema is a serious condition. It's a major cause of mortality and morbidity after, for example, conditions such as stroke. The same way that, you know, when you damage your ankle or your hand or anywhere in the body, that part swells. The same happens in the brain, but in the brain there is no enough space for swelling. So when brain starts swelling, the condition gets very dangerous. I think, let's see, I can show this first. So here you have a CT of a large stroke in the medial cerebral artery. And here you see the dark area is actually the area of edema formation. And this is a post-mortem brain slice showing a swollen brain. So this is brain edema. Unfortunately, we still don't have good treatment for brain edema formation, and the most serious types of brain edema, you know, what actually saves lives is surgical decompression, that you basically remove part of the skull and let the brain swell, and then when it goes back, you hope that the patient will survive, and that's how some patients actually survive. So we don't have a treatment. So discovery of aquaporine water channels on the interface between brain and blood opened this opportunity that maybe that's how water enters the brain, and what if we block these channels or remove them? And at that time, there were no selective blockers of these channels. So the question was, what is the mechanism by which these aquaporins are surrounded exactly or anchored to these specific membranes? And together with John Neely, was a PhD student at Peter Agger's lab, we worked on the anchoring mechanism. John suggested that there were some indications that already before that dystrophin complex was involved in the anchoring of aquaporin 4 to this membrane, to astrocytic membrane domains facing the vessels. And that was because of the studies in muscle. As you know, dystrophin is known from muscle dystrophies. So he identified a member of this protein complex, of dystrophin protein complex, called alpha-syntrophin, which is a PDZ domain containing protein. And the idea was that through interaction with between aquaporin 4 C terminus and alpha-syntrophin, aquaporins are anchored to these membranes. So we looked at alpha-syntrophin knockout mice, which were generated at the University of Washington in Seattle by Stan Froner and Marv Adams, other collaborators. And we looked at the distribution of aquaporins in these mice, and that was the picture that I jumped over. You see here, this is a wild type mouse with aquaporins, with shown here, but black dots in the astrocytic membrane. This is the endothelial cell. And in the knockout, we saw that aquaporins were not anymore concentrated here, but they were mislocalized to other membranes. So that was, now we had a model where aquaporins were not on the interface between brain and blood, and we could test our hypothesis. Is it how brain edema is formed through these channels? So if that's the case, then alpha-syntrophin knockout mice would be more protected. So we induced stroke in the mouse model together with Anish Bhardwaj at the Johns Hopkins. And here you see a wild-type mouse. The white area here shows the extent of stroke. We saw that mice lacking aquaporins in these membranes, they had not only less edema, but also smaller stroke size. And that was very interesting. Other groups, like in Jeff Manley, University of San Francisco, California, San Francisco, they also showed the same in aquaporin 4 knockout mice, that they are protected against edema formation in stroke models. So at that time, it was already proven that aquaporin 4 could be a drug target when it comes to formation of edema after a stroke, and also we showed it in other models such as hyponatremia. We also, or other groups in Alan Bachman's group, they showed that also other conditions which induce vasogenic edema, when where the blood-brain barrier is leaky. In those conditions, it's good to have aquaporins because then water can leave the brain through the channels, while in conditions where there is no vasogenic edema, it's not good. It's it's actually harmful to have aquaporins because that's how water enters the brain. So that's for the edema part. So that in the recent years, I have been more interested in the neurodegenerative disorders. As you know, there are several neurodegenerative disorders, Alzheimer's disease that Sasan works on, Parkinson's disease, ALS, and so on. And what is common for them is that there is a chronic progressive loss of neurons in certain parts of the brain. And there are also other things which is common, is that neuroinflammation is a hallmark of most of these disorders. So, and here, just to show another picture about showing inflammation or microglial activation in the Alzheimer's disease. This is patient with Alzheimer's disease. So there is an ongoing neuroinflammation in people with neurodegenerative disorders. And what is common when it comes to aquaporin 4 is that, at least in the animal models of these disorders, there is an increased expression of aquaporin 4, basically in all of them. In some of them, we have in addition a mislocalization of aquaporin 4. For example, this is mesial temporal lobe epilepsy, a study we did together with Ture Eid at Yale. So here you see the brain sample from a temporal lobe epilepsy patient shown here. As you see, normally aquaporins are supposed to be concentrated around the vessels and not so much in the other membranes. But in mesial temporal lobe epilepsy, the distinct labeling around the vessel is lost, but then aquaporin level is increased in other membranes. So what we found there was that there is an overall increase in expression of aquaporin 4, but they were not in the same, they were not anymore concentrated around the vessel. The same picture we have found in a mouse model of Alzheimer's disease, where we see that, I mean, here this with immunofluorescent, that red is a beta amyloid, which is accumulated around some vessels. Those vessels where there is beta amyloid, we don't see aquaporins in the associating membranes surrounding them, but we see an increased aquaporin 4 expression in other membranes. So a general expression of upregulation of aquaporin 4 expression and mislocalization in some conditions. So I will just talk a little bit about the project we had in Parkinson's disease. So one question that we asked, does aquaporin 4 play a role in Parkinson's disease? The first thing that we noticed was that substance nigra, the area which is affected in Parkinson's disease, it is among the regions with the highest expression of aquaporin 4, both around the vessels and also in other astrocytic membranes. So compared with the neocortex, you see that it's almost twice as much aquaporins in substance nigra. So that was one thing. The other thing was that we look at the mouse model of Parkinson's disease, widely used. It's MPTP model. It's a toxin that we inject systemically in mice or in rat. Is taken up in brain. It's converted to MPP plus, which is the toxic substance, and it selectively kills dopaminergic neurons. What we could see was that the high level of aquaporin 4, which was already in substance nigra, it was even more increased in mice treated with MPTP. So that made us curious that, okay, we see increased expression of aquaporin 4 in several neurodegenerative disorder, and whether it actually contributes to the morbidity and maybe also mortality, or whether it is protective. So you see here that aquaporins are not only increased in astrocytic end-feet surrounding the vessels. This is the MPTP treated mouse, and this is saline controlled, but it's also increased in other membranes. So one hypothesis was that aquaporin 4 might contribute to the neuroinflammation. As you know, in neurodegenerative disorder, we enter a phase of chronic inflammation in the brain. This is based both on measurement of inflammatory cytokines, but also post-mortem and also in animal models, where we see that there is a strong microglial activation that persists. And this microglial activation is partly due to neuronal damage, but what I haven't shown here in another slide, I forgot to put it here. There is a crosstalk between astrocytes and microglia, and they actually contribute to each other's activity. And since aquaporin 4 is expressed in astrocytes and not microglia, so the question was, does aquaporin 4 in astrocytes contribute to activation of microglia? And there are some theoretical reasons for this question, because we know that one of the mechanisms by which astrocytes activate resting microglia is release of ATP. And a colleague of mine, Alan Nagelus and Mark Trane, they showed that astrocytes release ATP in an aquaporin 4 dependent manner. So if it's the case that astrocytes activate microglia in an aquaporin 4 dependent manner, then it would also mean that the increased expression of aquaporin 4 that we see in neurodegenerative disorders, it actually contributes to the chronic inflammation that we see in these conditions. So we needed a model where we have not systemic, but local selective neuronal loss and neuroinflammation. And it's more or less the same model, MPTP, but this time we used intrastriatal injection of the active substance, MPP+. So when we inject it in striatum, the idea is that it is transported, the toxin is transported through dopaminergic axons, through retrograde transport to substantia nigra, and affects neurons. So here we will get a neuroinflammation, substantia nigra, far from the area where there is where we made the injection. And then we could see whether we find a difference between aquaporin 4 knockout and wild type mice, and our indicator for inflammation was expression of specific microglia activating genes. So this is just part of the study I'm going to show you. So first of all, we see that when we inject the toxin in striatum, we get neuroinflammation, both astrogliosis and microgliosis in the ipsilateral substantia nigra. And we see it both in the wild type and in aquaporin 4 knockout mice. When it comes to the genes, we see that astrocytes, this is GFAP, a marker of astrocyte reactivity. Astrocytes are activated in both wild type and knockout mice. There is slightly less activation of a microglia marker, but when it comes to the genes that are involved in the inflammatory phenotype of microglia, like CD14, TREM2, we couldn't see any increased activity in aquaporin 4 knockouts, while in the wild types there were several-fold increase. So, and we looked at many genes. I'm just showing some of them. That common was that basically none of the microglia activating genes that we look at, they were increased in expression in aquaporin 4 knockouts, while they were strongly increased in the wild types. So our conclusion was that in the wild type situation where astrocytes have aquaporins and their aquaporins is increased upon treatment with this Parkinsonian toxin, we get an increased activation of microglia, which again leads to dopaminergic cell death. While in aquaporin 4 knockout mice, only one of the genes was significantly increased, and the microglia in these mice, they were not so active. So we think that aquaporin 4 has a pro-inflammatory role. And some years ago, we together with Peter Agre, we looked at cerebral malaria and role of aquaporin 4. We could see that unlike what we expected, aquaporin 4 level was decreased in the mice with cerebral malaria. But more importantly, we could see that when we started treatment with the anti-malaria medicine 5 days after the infection, most of the wild type mice survived, while most of the knockout mice died. So this is, I think, one of the few studies where we show that aquaporin 4 protects against a condition where there is both brain edema and inflammation. And it is understandable, because if you imagine that microglia are important in the fight against parasites and other microbes inside the brain, and if aquaporin 4 plays an important role in activating these microglia, then it is logical that mice which do not have aquaporin 4, they have less activation of microglia and less resistant against cerebral malaria. So I think I stop here. I thank all my colleagues, and I wonder whether you have any questions.
خیلی ممنون. لطف کردید